Hi, good afternoon. My name is Ichiro Takeuchi. I'm from Nagoya University and RIKEN. I'd like to thank the organizer for this wonderful event. And today I'm going to talk about the statistical test for deep learning-driven hypothesis. This is a joint work with the following team members. So let me start from a motivating example of brain image analysis. So in brain image analysis, there are mainly two tasks. The first task is to make a prediction. Suppose that we have a trained neural network and given a medical image, test image, then we want to predict whether this patient has a cancerous tumor or not. This is a simple prediction task. But this is not sufficient. Doctor would like to know which part of the brain has the tumor. So we want to find some tumor lesion. So I'm going to call it knowledge discovery. So in today's talk, I'm going to talk about providing statistical significance measure for this knowledge obtained by deep neural network. And I'm going to provide a method to compute a p-value of this discovery. And I'm going to develop some statistical test for this knowledge. So therefore, I'm going to call this knowledge as a hypothesis. And since this hypothesis is generated by deep learning model, I'm going to call it deep learning-driven hypothesis. So there are multiple approaches for finding hypothesis for this brain image analysis. For example, we can use saliency maps, something like CAM or GradCAM. This is approach one. And we can simply apply image segmentation, for example, by using UNet. And the third approach is that, for example, if we use, for example, vision transformer, we can look at the attentions. This is another interesting approach. And the fourth approach is that we can, for example, detect the anomaly region by using generative models, such as diffusion models. So let's see one by one. So this is the first approach, saliency map method by GradCAM. So here, suppose that we have already trained, let's say, a convolutional neural network for predicting whether this is cancerous or normal. Given a test image, then we can compute saliency map, for example, by using GradCAM. Then, for example, if we pick up the set of pixels whose threshold is greater than, whose GradCAM values are greater than a certain threshold, we can define a saliency region. And we want to provide a p-value for this saliency region. In the second approach, we can just apply like image segmentation to this kind of brain images. So if we have already trained the UNet, for example, for image segmentation, we can obtain the object region and the background region. And we would like to compute the p-value for this object region. The third approach, for example, we can apply a vision transformer to these brain images. Suppose that we already trained the vision transformer, and then we can show the attention values. Again, by selecting a set of pixels whose attention values are greater than a certain threshold, we can define an attention region. And I want to compute the p-value for this attention region. The third one is anomaly detection by, the fourth one is anomaly detection by diffusion model. Suppose that we already trained a diffusion model only using normal cases. So therefore, the diffusion model will produce normal image only. So if we provide this brain image that contains tumor, it will generate a corresponding normal image. And by comparing this image and this counterfactual image, we will be able to identify anomaly region. And I want to compute the p-value for this anomaly region. So I'm going to talk about how we can compute these p-values. So this is the outline of the rest of my talk. In the part one, I'm going to discuss how we can formulate the statistical test problem for deep learning-driven hypothesis. And in part 2, I'm going to introduce so-called selective inference. This is a new type of statistical inference that is particularly useful for data-driven hypothesis. And in the part 3, I'm going to talk about how we can use selective inference for deep learning-driven hypothesis. So part 1. So let me formulate a statistical test for deep learning-driven hypothesis. And I'm going to use an example of saliency map approach here. So let's say we have trained CNN already. Given a test image, this provides a saliency map, for example, by using a GLACAN. By setting a threshold, we can select a set of pixels whose GLACAN values are greater than a certain threshold. And I'm going to call it the saliency region. And in order to test, in order to quantify the statistical significance of this saliency region, we can think about a reference image that is known to be normal. And we just pick up the exactly same region from this reference image. Then we can compare the distribution of the pixel intensity between this and this. And for example, we can conduct two sample tests, something like a t-test. Then we will be able to get the p-value. So this is one of the formulation, one of the statistical testing formulation to quantify the statistical significance of this saliency region. Here, now we have a brain image that contains a tumor lesion in the upper area, and in the bottom we have the a brain image that does not contain tumor lesion. So what we expect here is that in the upper case, we expect a small p-value because we want the method to declare this is statistically significant. And on the other hand, in the bottom case, since this is a normal image, some lesion is perhaps just an artifact obtained from the noise. So therefore, we expect that the p-value here to be large and saying that, indicating that this is not statistically significant. So let's do that now. So now we have a brain image that contains a tumor here. And we compute the saliency map and the saliency region and provide the reference image that is known to be normal and pick up the same region. And just compare the pixel intensity distribution of these two. And the result is something like this. Now, for simplicity, let's forget about this saliency region is obtained from the neural network. Just comparing the distribution of the pixel intensity, we will have p-value to be 0.00, indicating that this is like truly positive, indicating this is statistically significant. The difference are statistically significant. This is fine. This is what we expect because we want the method to tell that this region is something special. But the next case is problematic. Now, we have a brain image that does not contain tumor lesion. So at this, in this image, we want a method to say that this is not statistically significant. But if we forget about the fact that this saliency region computed from the neural network and just comparing the pixel intensity of this saliency region and this saliency region, the result is again p-value to be 0.00, indicating that this is highly statistically significant. This is actually false positive detection. So there are something problem here. So now let me formulate this problem a little bit more concretely. Now, let me call this image as an n-dimensional vector x. And we feed this image into the trained neural network, and this will compute the saliency region. And we have the x reference. This is a reference image, and we can pick up the same region in the saliency region here. And we conduct a two-sample test. So let me formulate this mathematically. Now we assume that the test image x is the combination, like consists of the signal part S and the noise part epsilon. And now let me assume that the noise part epsilon follows normal distribution, and the reference image same. X reference consists of the signal part and the noise part. And now let me call this trained neural network as an algorithm A. Now algorithm A like maps an image X into a salient region, MX. So this is just a simple formulation. Now, hypothesis testing problem is formulated as shown here. The null hypothesis here is that the average signal in the saliency region in the test image is the same as the average signal in the corresponding region of the reference image. So this is like null hypothesis. And the opposite alternative hypothesis is its opposite. They are not equal. And now our reasonable test statistic would be just the difference between the average pixel intensity in the test image and the reference image. And in this formulation, we can just compute the p-value by looking at the probability that the this difference is greater than the observation. So this is like a simple two-sample testing formulation. But it is important to note that now the hypothesis we are interested in is selected by a deep learning model. So now there are a lot of hypotheses to be considered. Since we now have 5 times 5 pixel, so now the all possible hypotheses that we can consider is 2 to the power of 25. Actually 2 to the power of number of pixel hypotheses we are considering. And the deep learning model that is well trained can select the best one from that. So therefore, it can be interpreted as a like very ultra large scale multiple testing problem. Now, deep learning model are trying to select the smallest p-value case from all possible cases 2 to the power of the number of pixels. So therefore, we cannot like just pick up the smallest p-value in the scenario of multiple testing. So it is indispensable to collect the selection bias caused by the selection conducted by the trained deep learning model. So this is the source of the some weird situation, even for a brain image that does not contain a tumor lesion, the p-value tend to be very, very small. So if you are somewhat familiar with multiple comparison, this is like a traditional problem. But in the context of traditional multiple hypothesis testing, only a handful of tests are considered. Something like we are now conducting 3 tests, so therefore we have to collect the multiple comparison issue. In the context of genetic data analysis, that starts from around 2000, many people start to think about large-scale multiple comparison problem with tens of thousands of hypotheses. But now, what we need to consider for the deep learning is that the number of hypotheses is 2 to the power of number of pixels, which is much greater than this situation. So therefore, we have to think about something new. And this selective inference, that is the main key point of this talk, is the statistical inference for data-driven hypothesis. So this is a very simple illustration of the selective inference. So the key idea of selective inference is to consider only the cases where the same hypothesis is selected. So remember that in the statistical inference, we consider many parallel worlds. And we are only in one of them. We observe the data and then applying algorithm, hypothesis B is selected. But we consider that in other parallel worlds, other data are obtained, and the algorithm is applied, and other hypotheses are selected. In the selective inference, we only focus on the set of parallel worlds where same hypothesis, in this case, hypothesis B is selected. So we don't consider these parallel worlds. We only consider the case where the same hypotheses are selected. So intuitively, by considering only the randomness where the same hypothesis is selected, we can forget about the selection bias issue caused by the deep learning model selection. So this is another view. Let's see, this is a data space. In our example, this is an n-dimensional space. This point indicates an image with n pixels. And if we apply one image to the AI, deep learning model, particular hypothesis B is selected. And if we apply other image in the data space, of course, another different hypothesis could be selected. But here, we focus on, in selective inference, the set of images where same hypotheses are selected would be considered. So that means that in the data space, we don't consider the entire data space. We only consider a subset of data space from which the data point generates the same hypothesis. By doing that, we can remove the selection bias, multiple comparison issue, even when the number of hypotheses are 2 to the power of number of pixels. So what we do mathematically that, so this is the ordinal definition of the p-value. P-value is the probability that the difference is greater than the observed one. This is the ordinal statistical significance. On the other hand, in the selective inference, we consider conditional statistical significance. Here we have a condition part. The first part is the same. The difference is greater than the observation. And under the condition that same saliency region is selected. So by considering this conditioning part, we are doing something like this one. And technically, we need to condition on other things, something like sufficient statistical nuisance component. But anyway, conditioning is an important part of the selective inference. So let me talk about a brief history of selective inference. The notion of conditional inference itself has long been used in many problems and known in the literature of statistics. But in around 2015, Lee et al. First proposed the computationally tractable selective inference method for lasso problem. Inspired by this work, a particular type of selective inference for various other feature selection methods were developed. But selective inference has known to be very useful, not only for the feature selection, but other type of problem. And various extensions have been made to the computational aspect of selective inference. And I'm also working on that part and making it applicable to more complex selection, something like deep learning models. So it looks something like this. And this is a data space, n-dimensional real space. A point here indicates an image. And we consider a test statistic direction. This is selected by a neural network. And this is the actual image we have here. And in the test statistic direction, we can have this kind of sequence of images. And in this test statistic direction, we have the distribution. And the ordinal p-value is something like this. Like the probability that more extreme situation arises than the observed one. This is the ordinal p-value. And what the selective inference does is as follows. Now, we only pick up the case where same selected hypotheses are selected. In this orange region, suppose that the same set of hypotheses, this saliency region is selected. And focusing on particular case where the same hypotheses are selected, and the selective p-value itself is defined as follows. So we don't consider the case where different hypotheses are selected. By doing that, we can forget about the multiple testing issue or selection bias issue. So these are the difference between the ordinal p-values and the selective p-values. If we can compute this, then we can compute the exact p-value, even that hypothesis is selected from the neural network. So for example, when we apply selective inference to the saliency map problem, this is fine. This is true positive case. But in the case where there are no tumor here, naive P tells that this is highly statistically significant, but the selective P tells that this is not. This is actually because the neural network selected something seemingly different parts. Okay, so now the problem is that how we can compute this kind of orange lesion and this selective p-value in the case of neural network. Our key idea is that many components in the deep learning model can be represented by a piecewise linear component, or can be approximated by a piecewise linear component. For example, if you look at the Leroux activation function, this is clearly piecewise linear function. Max pooling, upsampling, convolution operation, those are all like piecewise linear operation. So that means that, for example, this horizontal axis indicates an input image X, and the vertical axis indicates the GradCam value. These are represented by this kind of piecewise linear function. In this case, we can check which part of the pixels are selected can be obtained by checking a finite number of intervals. So therefore, if the neural network can be represented or approximated by a piecewise linear function, we can exactly compute the set of images whose which is same hypothesis is selected. And actually, in the implementation part, this can be done by a trick called auto conditioning, which is in spirit similar to like auto derivative. Just by computing the forward step of the neural network, we can find the lesion. So, and we have already developed a nice implementation framework in the TensorFlow. So without any implementation cost, we can conduct a selective inference without any additional implementation cost. So this is a result of the approach to segmentation. Now this is a U-net, although it doesn't look like U, but anyway, so this is a trained U-net, and the segmentation result is obtained. And naive p-value and selective p-value are both 0 for the case where there is a true tumor. But in the case where there are no tumor, naive p tells it's statistically significant, but the selective p does not. So this can nicely detect the true negative situation. The approach tree, this is a vision transformer. Attentions are obtained like this, and the attention regions are defined as follows. And in the negative case, selective p is still high. This is different from the naive p. And this is like a diffusion model case. Now, remember that we have trained the diffusion model only with normal cases. Therefore, when we provide this brain image, then it will produce a counterfactual normal image of this patient. And by comparing this and that, then we will have the anomaly lesion. This is this type of anomaly detection approach has been studied very intensively, but we can compute the p-value for that. Now, selective p-value is very small in the case where there are tumors, but in the case where there are no tumor, naive p is still small, but the selective p is high. So, but this is just an example, but selective p-value is theoretically guaranteed to provide valid p-value when the hypothesis is selected. So this is a summary, and that's going to be the end of my talk. Thank you so much.
Thank you for a great talk. So is any question, comments from this?
Thank you for the talk. How sensitive is your calculation with respect to the reference image?
What is your question? Like we just pick up?
Well, I'm saying it seems to me that your proposal is very sensitive to the choice of the reference.
Yeah. So yes, of course, you are completely right. So we can select multiple reference image and thinking about the difference with the average normal image and this test image. So like, yeah, you are completely right. But we can, in order to remove the sensitivity, we can pick up the multiple difference images and compare the average with the test image, I think.
Okay, thank you.
Thank you.
So is there any other question, comments? Yeah, so yeah, I have one. So you mainly consider the trained AI model. So I'm interested in like a possibility of extend your framework to the more like a model including training. So is it possible in future also?
Yeah, that is a very important direction. But unfortunately, conditioning on the training process itself is at this moment, it's very, very challenging. So now, therefore, I now focus on the inference after the trained model. Yeah, but that is a very important direction. Thank you so much for the comment.
Yes, thank you. Okay, so it's almost time. Okay, so please thank speakers again.
Thank you so much.
Okay, thank you for the introduction. I'm Naoto from the University of Tokyo and also RIKEN AIP. My talk is about how deep learning is advancing remote sensing. And this is the contents of the joint work with many colleagues, including those listed here. So our group is working on the intersection between computer vision and remote sensing, and our research topics can be broadly categorized into those three topics. And one is how to get high quality image acquisition by performing inverse imaging problems like denoising, super resolution, or compressive sensing. And the second topic is image understanding. So remote sensing images are like this, as shown at the middle. So main tasks include the semantic segmentation and also 3D reconstruction from those image data. And based on these techniques, we are also working on solving social issues like through disaster management and also environmental monitoring. So remote sensing, please look at the bottom left-hand side. So here is the comparison of images having exactly the same size in terms of pixels, comparing with the image taken on the ground and the remote sensing images taken from airplane, which is a relatively high resolution image. But as you can see, the objects are really small because remote sensing is the technique to capture the image from airplane or satellite. So the sensor is far away from the objects. So objects are very, very small. So spatial resolution or other resolution like spectral resolution, temporal resolution matters a lot, but due to these constraints of the hardware or platform, there is no perfect data. So the data is always something incomplete compared to what we really want. And also each project is designed for specific applications. So each satellite has very different specification of the sensor design, which makes the analysis also challenging. And another challenge is lack of training data, which is often the case for many domains. So annotation of every single pixel for this kind of very complex things is very time consuming compared to ground shot images. And also some ground truth is only available by field survey, which costs a lot as shown on these photos. Also, another annoying regulation is that very high resolution satellite data, there are a lot of data, but there are regulations on high resolution satellite data accessibility due to the security reasons, which slow down the construction of publicly available data, so which is very different from the computer vision community. So our group is working on addressing these challenges, and today I would like to introduce some of our activities through three topics. The first topic is about image understanding to enable automated mapping for everyone. Everyone means everyone from any place on this planet. So last year we have published the benchmark dataset, the first dataset for global sub-meter level land cover mapping, which is named Open Earth Map. So due to the reason I explained before, there was no public data set that is covering various regions on the globe when it comes to very high resolution imagery. And we constructed this data, and the model trained on our data works pretty well for any images taken from any place on this planet. And this is just an example taken from Tanzania. And the model predicts something like this. So for every single pixel, even very, very small trees can be identified in a quite stable manner. So the problem in our domain was that there are many, many benchmark data sets. People create benchmark data to do research, but as shown on this world map, except for these light blue colored, other colors are high-resolution images of benchmark data sets for designed for building detection. And as you can see, these other colors, except for light blue, are coming from mainly US and Europe. So whatever we annotate these images, the trained model works really well in US and Europe, but it doesn't work in Asia and Africa and also South America, which is not acceptable for us. So our colleagues are from also Africa and South America, so we collected the publicly available data additionally to make the number of images balanced among different continents. And the data set is not really huge, but 5,000 images covering various regions. In this domain was the first data to cover so many countries for very high resolution images. And here's an example of annotated data, so pixel-wise labeling. Very, very spatially detailed annotation is required, and how to make annotation consistent was quite challenging. And this very high resolution semantic segmentation is an obvious task that everyone wants to do. So there are some competitors like German Aerospace Center or US Department of Defense and some other university and institutions. And our data sets has the advantages over other existing data sets in terms of geographic diversity and annotation quality, which is measured by the number of segments. And this difference makes difference during the inference. So here's an example of how the prediction are. So from left to right, input image and reference data, map data, and our prediction result. And the 3 columns on the right-hand side are the prediction result of the models trained on existing benchmark data sets. And as you can see, by the way, the neural network model is the same. UNet using efficient net B4 as the backbone, and just the difference is the data. So as you can see, prediction results are quite spatially detailed and accurate, similar to the reference. And we also confirmed it's working quite well with the numerical evaluation. So the model can work as a kind of off-the-shelf model for many applications. And one application is the disaster response. And this is the example applied to satellite images pre and post disaster for the earthquake in Turkey and Syria last year. And you can see that destroyed buildings and roads can be identified. But by just comparing these bitemporal map data, we can get a change map. But usually the satellite capture the image not always off 90 angle, but having a bit of pointing angle, which makes the image looks a little bit different. So one image, sometimes the facade of the building is visible, but in other images, it's not visible. So such a slight spatial misalignment can lead to lots of false positives if we just perform change detection by comparing those classification maps. So if possible, we want to also make the model to be trained on the paired pre and post change images with change masks, but it's very, very expensive, more expensive than what we have made. So the one strategy is that to create the synthetic data to make a very realistic remote sensing images. And this work is done led by Mr. Johnson, a PhD student in our group. So he built up the remote sensing image synthesis tool based on procedural modeling, which is a standard technique in computer graphics, together with generative models to increase the diversity of textual information. And we built up the data for a bit more advanced tasks like building change detection. And also we are interested in estimating height information, not only semantic information, but some digital elevation model is also our scope. And with real data, it's very expensive, either by stereo vision or LiDAR measurements. It's expensive and it's very difficult to collect diverse data set covering different characteristics of geographic regions. So here are examples how this synthetic data can help to improve. On the left-hand side is showing that using this synthetic data in addition to real data can help to increase the performance when there is a domain gap between training data and testing data. And the right-hand side is the similar results, but for another task, building change detection. So together with the real data, we have confirmed that for real benchmark data sets, this change detection synthetic data could boost up the performance for building change detection. We are working not only just for benchmarking. Our motivation is real application in helping and supporting disaster response. And in Japan, this year, January 1st, there was a big earthquake in Noto. And here is an example of the building change detection result from left to right, pre and post disaster aerial images. And the right-hand side is the building change detection result obtained by the model trained on this synthetic data as well as open earth map that I presented. And you can see that the bottom images are zoomed in images. So our model could robustly identify the destroyed buildings as well as land cover changes with less false positives compared to the existing models or just the post-classification comparison. So our interest is to estimate some semantic information, and also we are interested in height estimation, particularly molecular height estimation when it comes to deep learning-based approaches. But this height information is just actually 2.5 dimensional data. It doesn't have 3-dimensional information. It's just height, and we never know how is the structure under the top canopy of the trees, for example. So our ultimate goal is to convert remote sensing data into full 3-dimensional models. And towards this direction, we are also working on the intersection between machine learning and computer graphics. And this is another example, deep tree sketch. This technique, this is led by Mr. Jihao Liu, another PhD student. So input is a sketch, just draw sketch, and a system output, very, very highly detailed 3D structure of the trees, as you can see from these examples. And this technique is composed of machine learning and, again, computer graphics techniques, so-called procedural modeling. So the first step is just the sketch image is just 2D, very simple sketch. And we can regard it as a graph structure by considering the intersection of trunks and also branches as node and the branches as edges. So progressively, we can perform depth estimation for every single 2-dimensional node to get a very rough structure of this tree. And after that, procedural modeling, which is a rule-based algorithmic approach, which is common in computer graphics, can make branches grown up and make the final reconstruction result, which is quite realistic. So the first part is this is a tree graph. This is named tree graph prediction network. It has two encoders. It's a quite simple structure and composed of local feature encoder and global graph encoder. The first one accepts just the 2D coordinate of the current tree visited node, and the latter one accepts the entire graph structure, which is represented in the matrix. And these are based on MLP and also graph convolutional neural network. And then output is just a very scalar value of the depths of the currently visited node. And it works in a progressive manner to estimate the depth information. And the training data, again, training data matters. And here we, again, used both synthetic data and real data. And this kind of technique can be applied to the real photographs. For example, we can just trace main branches for a given photographs, and then the system can output very realistic 3D reconstruction results. And we are currently targeting, this is only single tree, but what we are targeting is to do this level of detailed 3D reconstruction for much larger, very large scene of the city or country, or ultimate goal is the entire globe. And a bit more application disaster response related topic is, again, lack of training data. The disaster is quite rare, and collecting real data is really not possible to collect enough samples. And our group collaborates with space agency and also disaster management agency, and whenever something happens, we apply image processing or machine learning techniques to see what we can do and what is the problems to solve. And here's one example. There was a big typhoon named the Hagibis 5 years ago, and almost half of the Japan was attacked, and flooding happened everywhere. And this is an optical imagery pre-disaster taken by Sentinel-2, which is operated by European Space Agency. And this is the next one. This is the post-disaster optical imagery, which is, how to say, covered by cloud, and we cannot see the ground. So unfortunately, optical imagery is really intuitive and useful for extracting semantic information, but unfortunately, it's useless because we cannot see the ground. So the only sensor that we need to rely on is another modal data based on radar, which is called synthetic aperture radar, which is an active sensor emitting microwave from the satellite and then measure the backscattering signal from the ground surface. So these brightness is related to the structure of the surface. So if the surface is, for example, water, the microwave just reflects and never come back to the sensor, so it's dark. And if there is elevated objects like buildings, and then the microwave hit on the ground, reflected, and then reflected again on the facade of the building, come back to the sensor. So that's why it looks brighter. And since it's using microwave and active sensor, we can get the image without sunlight, and also even with cloud. So this is a pre-event, pre-disaster SAR imagery, and this is a post-disaster SAR imagery by switching pre and post. I hope you can see the difference in intensity. And by analyzing these pre and post-disaster images, it's possible to detect a flood extent map. But when we discuss it with disaster management people 6 years ago, they ask, okay, this is better than nothing, but is it possible to estimate inundation depths? So whether flooding inundation was just 50 centimeter or 3 meters, they mean totally different levels of damages on the buildings. And also when debris flow happened, where the soil eroded and where is the sedimentation happen. This kind of information is necessary for disaster recovery during the recovery process, because how many trucks we need to send to this place, or it's up to the amount of soil. So such three-dimensional, or let's say 2.5D dimensional information was not possible to get from only simple 2D remote sensing images, but at that time we are working on estimating inundation depths as well as the topographic deformation due to debris flow. So this was the mapping result of the inundation depths that we provided through the real actual disaster response activities obtained from synthetic aperture radar imagery together with deep learning and numerical simulation. So compared to the reference data, there is in Japan, disaster management is very important. So the Geospatial Information Authority of Japan usually publish this kind of inundation depth map based on human visual interpretation of the aerial photographs. And we confirmed that, yeah, our result was quite consistent. Let me explain what is the trick behind. So it's a kind of combination of remote sensing, a numerical simulation, and deep learning. So from remote sensing images, we can get binary change information, but unfortunately, that's all. If we have LiDAR sensing or some other measurements focused on measuring height, it's another story. But if we have only 2D optical or SAR images, binary change information is what we can get, which together with some noise. And there is another research discipline based on numerical simulation, and they can simulate something based on some model and numerical simulation. And by changing input data or parameters, they can simulate many scenarios, how it looks like. But it doesn't fit with the reality, because for precise numerical simulation, for example, for debris flow simulation, they need a precise location of the starting points of the debris flow, which is not available right after disaster. So simulation result doesn't match with reality, but it can get some sort of 3D change information. Remote sensing can reflect the reality, but we cannot get 3D information. So we try to combine the advantages of these two approaches through deep learning by considering simulation data as training data, by constructing regression models using binary change and topographic data as input, and then output maximum water level or topographic deformation map. And so once this regression model is trained, we just need to input binary change information together with topographic data, which is available in this country. And here are a bit more results for more challenging task, which is debris flow mapping. And the middle example is the binary change detection with conventional image processing approach. And the right-hand side is the topographic deformation estimation result, where blue and red represent the erosion and also sedimentation, respectively. And we confirmed, together with some LiDAR measurement, we have confirmed that these trends are quite consistent with what really happened. Okay, so the last topic is about image acquisition. So as I mentioned, remote sensing is acquiring images far away from the target, so spatial resolution matters. So if we can see more spatial details, and then deep learning can do a great job. So we want to acquire high resolution images, but there is always limitation in hardware. And the image fusion based super resolution, or it's also called guided super resolution, is it's a super resolution technique by fusing two different images, which have some sort of trade-off between, for example, in case of pan sharpening, it's a fusion of high-resolution panchromatic imagery and lower resolution multispectral data to reconstruct the higher resolution multispectral data. So probably you have seen some satellite images somewhere, and these images are almost all pan-sharpened images. These are not real measurements. So this is really necessary in Earth observation. And another more challenging task listed at below is the cross-modal super resolution. So when the modality, the data modality is different, less correlation exists, so it's much more challenging. And the supervised learning, of course, can achieve great performance, but the problem is, again, in our domain, we cannot get ground truth, or we cannot get training data. We cannot get high-resolution multispectral image. So unsupervised approach or self-supervised approach is the key in our domain. And conventional approaches using only input image is basically based on optimization. And the people try to design some regularization terms to exploit the image prior, like sparsity or smoothness or non-local self-similarity or low rankness, and so on. So later, inspired by deep image prior, our group is working on methods that regulate the inductive bias of the neural networks. So it's a kind of characteristics of the architecture design itself. For example, if we stack convolutional filters, such network can easily fit to smooth patterns. Or In our work listed here at the bottom, deep attention playa is if we stack attention, self-attention mechanism or attention layers, it has a good capability to fit to non-local self-similarity, which is quite important in image processing. So, which is useful for image inverse problems. So instead of optimizing the target imagery, we can change the task to optimize the parameters of the networks, just using input images. And lastly, let me introduce this mutual modulation. This is led by Xiaoyu Dong, another PhD student in our group. She worked on this cross-model super resolution without using external data. So basically only using input image. And here is an example for RGB imagery, high resolution, and a bit lower resolution source image. And this is the ground truth. And this P2P is one of the existing approach. And I hope you can see there are some artifacts happening here, and which is injected because of the patterns coming from guide image. So we want to enhance the spatial resolution, but when the modality is different, we don't want some spatial details that is not really correlated, that is not relevant for the source image. So how to achieve both is a bit of challenge if we focus on the approach without using any external data. And what we proposed was a quite simple approach to plug in spatial filtering at the middle of the feature extraction, which is named mutual modulation. So it's a spatial filtering is a very, very basic elements in image processing. So after extracting features, we compute the spatial filter kernel weights by calculating the correlation between the current interested pixel and the features of the neighboring pixels, but which is taken from another modality's features. And we can do the other way around to create two different spatial filtering. So there is source to guide and guide to source modulation. And each of them has different role. The first one has the role to enhance the spatial resolution of source image, which has lower resolution. And the latter one has the role to remove unwanted spatial details in the guided, the features extracted from guide image. So for various combination of modalities like thermal data or digital elevation model and depth images, we have confirmed some stable and high performance of our approach, even compared to supervised approaches. So this is the summary, and yeah, that's all of my talk, and thank you so much for your attention.
What a great talk. Are there questions from the audience?
So I really like the binarization from simulation stuff that you did. I was wondering, is there any issue like when the real thing happens, right? It's not based on simulation. Like how do you incorporate the reality with the thing that you learned from simulation? And do you do something additional to make sure that you don't get some biased things?
Thank you. So thank you for the question. And this is a very good point. And currently, this concept works when simulation data is really realistic. If it's far away from the reality, it doesn't work, unfortunately. And in case of flat simulation, the diversity is much less. So if it's quite correlated with the topographic data, but the debris flow is quite challenging, and depending on the topography and also what kind of soil exists under the ground also affect what kind of debris flow happen. So it works. We tested with many different areas, and also, how to say, separate training area and test with other regions, and flat inundation depth estimation works, but debris flow mapping doesn't work. So I believe what you suggested is needed. And so somehow fine tune to the test area by getting some sort of information from test area. So without any information from the test area, it doesn't work, unfortunately. Yeah, thank you so much.
Are there further questions? Okay, if not, let's thank Naoto again for the great talk.
Okay, thank you so much.
Thanks for the nice workshop, and thanks for the organizer for inviting me. So what I'm going to share with you is some of my recent experience on using deep learning for some of the biological problems, right? So we all know that the pre-trained language model for natural language has made a huge kind of improvement in terms of our ability to represent natural language, and also tailor it for lots of downstream tasks, right? In terms of classification model, regression model over text, and also be using that for question answering. But the biology is just speaking very different language, and here is just a glimpse of the complexity of the biological system. If you zoom into a cell, you will see that different type of molecules are there. From the genetic materials in the DNA, you actually need to have these RNA to transcribe that genetic information. And RNA is going to be translated into protein. And protein is going to fall into three-dimensional structure, and it has dynamic property as well. And then the protein is going to interact with other protein to form a very complex network within the cell, and regulating back to the expression level of RNA, things like this, right? And each cell is going to communicate with other cells and receiving and sending informations to form more complex, high-level kind of systems, right? If you look at the biology, it's really a multi-scale system like this. From the finest grade or the lowest scale, it's going to be those actins and molecules, right? By themselves, they have their structures. They're going to interact for kind of molecular interaction networks, and there's a cell-cell interaction, and to carry out the function of particular organ and tissues. And this organ and tissue within the particular species is going to coordinate and carry out the function of that particular organism. And not to mention that you have different kind of organism or people communicating with each other, and form a very complex social network spreading diseases and things like this, right? And actually, there's many different ways of measuring information from different kind of scales of the complex system, including measuring the sequence of the DNA, and then the sequential information for the protein and three-dimensional structure, as well as looking at the expression level of the entire cell, right? Single cell gene expression, not to mention images and some other clinical diagnosis technologies. You collect lots of information about different kind of organisms, right? So if very often we want to build some model for making predictions about problem arising from different scale. Klaus just talked about how do we actually obtain some of the dynamic information about a particular molecule, right? And there's also a question about how do we actually make prediction about the responses of a particular cell to some treatment or drug, right, to find out new kind of targets. There are also questions about how do you actually make prediction about image from the tissue, and how to actually provide the right treatment for that particular kind of tissue you have seen, right, or the type of the disease you have seen, right. So actually many problems arising from biological system, healthcare or life science is some kind of problem with high dimensional inputs, but the amount of labeled data is really small, right? For instance, if you want to actually look at a new disease, for instance, immune related diseases, and what you want to do is to find out the so-called target. What will be the key kind of protein you want to target in order to, for instance, avoid a disease or eradicate a particular disease. And then you actually want to identify within this complex network of tens of thousands of proteins or other molecules, which are the important nodes. Sometimes you also need to look at a combination of not just one node, but a pair of nodes. Then you also have this combinatorial space you want to search over. Once you identify, the target or the critical node in this network, so that particular node again is a molecule like a protein, right, or other type of molecule in the system. Then you want to design a drug which is either small chemical kind of molecules, or it's going to be a protein, right? When you want to design a protein drug, for instance, antibody drug to it, again, you want to design, first the sequence information for that particular protein. So say you have a protein of length of 100, then each position of the protein can take 20 possibilities. You're also designing in a high dimensional space, 10 to the, essentially is 10 to the power of, 100 to the power of 20, right? And very often you are limited in the amount of, money or time to do the experiments. If you're going to measure all possible, combination you can have for designing that particular protein, that would cost a lot, right? So you only collect small number of data points in this space, and it's very sparse comparing to the gigantic space you are designing that particular molecule, or the amount of patient kind of a sample, or the type of the number of cells you can actually test it on is much more smaller comparing the space you can explore and put up the cell to collect the information. So the question is, how can we, in this case, learn some representations, right? Or build some kind of models such that will be helpful for us to deal with this kind of small data regime, right? So in the kind of general artificial intelligence kind of regime, people are now using this kind of framework or paradigm of foundation model quite a bit nowadays. So essentially, what people find is there are lots of unsupervised data out there, right? For instance, there's lots of text from internet, there are lots of images from internet. What you're going to do is to first, build some kind of self-supervised pre-trained model based on those unlabeled data sets. You can think about many different ways you can construct this self-supervised learning, either masked language model for recurrence of the missing information, right? Or generative models where you want to predict the next token or next words based on partial sequence inputs. Or you have contrastive learning comparing two instances of the same example, and you want to actually differentiate it from other instances, things like this, right? Then once you have this pre-trained model, typically based on billions of input instances, and train with maybe hundreds of GPU for a long time, and then you get this representation. And when it comes to downstream tasks, either for building sentiment analysis or even question answering, you only need a small number of examples to just fine-tune this type of pre-trained model in order to do well in downstream task. Nowadays, what people find is this pre-trained model is indeed actually in many downstream tasks is helping you to boost the performance by quite a bit, right? And there's also interesting kind of observation that some tasks, the performance will actually improve as you pre-train a larger and larger model, right? So the question is whether this particular paradigm can be actually used in biological problems, right? And if this is a good paradigm, what kind of data should we use to pre-train these models? And what kind of downstream tasks can benefit from such pre-trained model, right? Okay, so in fact, the success of this type of pre-trained model actually at least has three very important factors. One is a huge amount of unsupervised data, right? Particularly sequence data, image data. The second factor is these big transformers, right? Being able to actually learn representation from this particular type of a huge amount of data. So the data is typically billions of data points and billions of parameters also for the transformer. And the third factor is you have lots of computations actually being used to train these models, right? So over the past 2 and 3 years, I've been exploring sort of building some kind of foundation model for the biological problems, right? So when I look at the space of data first, the first factor, right? So it seems that there are quite a lot of unsupervised data already in the biological space. At least, people have done lots of projects in biology, and then cumulatively, when you put together those data, yeah, some of the data set has already reached or close to the internet scale. For instance, if you look at the protein sequences data out there, there are already billions of protein sequences out there. So thanks to the increasing kind of efficiency about sequencing and reduced costs about sequencing, many protein actually can be obtained by just taking some sample to sequence it. You discover new proteins, right? And similarly, there are lots of single cell RNA-seq data as well, right? So people are able to take some cells and being able to measure the number of genes being expressed in each individual cell. So now there's also hundreds of millions of cells being sequenced, right? And each cell contains 20 to 30K genes, and altogether you get lots of data as well, right? So the goal is to actually have some kind of ability to absorb the information in the data. Yeah, you build some kind of pre-trained model based on this huge amount of data, and then use the presentation for downstream task and see whether you get actually some benefit or improvement based on this pre-trained model, right? So the entire kind of family of kind of transformer model you want to build is actually crossing different kind of modality data. There's a DNA data, RNA data, and protein data, and single cell and seq data, reflecting the complexity and the multi-scale nature of this biological system, right? So today I'm going to mostly focus on some results and explorations in protein, foundation model, and also mentioned about the pre-trained model for single cell and seq as well. And you get the chance to see a little bit by pre-training, actually you get quite a lot of improvement on this data, right? First, for protein, so actually it's relatively easy. You can make a mapping between protein sequences and to natural language. So protein consists of, it's going to be a string with 20 different possibilities in each location. The vocabulary size is just 20, right? You can think about an amino acid is just like a lactose in English or token in your natural language. But protein is like a sentence, right? A sequence of tokens. And there's going to be protein-protein interactions, and protein is going to form networks, and it's like a paragraph. You have an intricate kind of connection between different sentences in a paragraph, right? So protein, of course, is not going to stay as a string. It's going to fold into some kind of three-dimensional structure. It is a dynamic structure, and this dynamic structure is actually determining the function of that particular protein. And protein is going to function by binding or interacting with other protein and other molecules. And there's a complex network carried out the function. So what we want to do is to at least take these billions of protein sequences out there and apply this pre-training kind of model framework and see what we are going to get in terms of this pre-trained embedding for structure prediction, other property prediction, as well as generation as well. Right? So what we did is actually train the 100 billion parameter model for these protein sequences. So in this particular model we trained, we actually mixed together two kind of objective functions. One type is mass language model, which asking the model to fill in some of these blank spaces in the middle of the sentence or the middle of the sequence. And the second objective we have as a mix is going to be just generative model, asking the particular model to also recover in the second half of the sequence, given the first half, right? The next token kind of prediction. So the parameter is going to be shared between this mass language model objective and also the GPT. It's just that in the example you input into the model, you have sometimes asking it to recover the mass position. Sometimes you're asking to actually recover the second half of the sentence or generative pre-training, right? By actually pre-training such a big model, the same model has two type of ability. One type of ability is using this embedding for building some of the downstream classification prediction model, and we call understanding ability. The second ability is having the model to generate new sequences, right? Generative model, right? Allowing you to explore the space, right? So essentially, you can think about this pre-trained language model is able to support quite a variety of tasks on the top, including the generative model, the understanding task I just mentioned. You take the input sequence and then you get embedding. You can perform the pooling of all the embeddings and to build some classification regression model about an entire sequence. You can also do more complex tasks such as predicting the structure of the protein, right? I'm going to show you some example and comparisons. The second type of task we want to do is the generation, right? Generating new protein sequences which resembles natural protein sequences, but at the same time have some variations such that you can explore these protein spaces and find some new designs and interesting new kind of proteins, also conditional generations, right? So interestingly, such a large model hasn't been done in the sort of the biological kind of community or in the medical community yet. And some of the earlier model is actually also being trained in internet companies such as Meta or Salesforce. One reason being that it requires quite a bit of understanding about the transformer deep learning architecture and quite a lot of engineering computing resources to get it working, right? So comparing to the previous, second largest model out there, which is the Meta, 15 billion parameter. So we are sort of almost doubling the number of layers, right? And then the embedding dimension is also doubling, and in total, it has 8 times more parameter comparing to the second largest model out there, right? So the model is changed just by some of these mass language model kind of objective function and generative kind of objective function. But after you finish training the model, what you will find is the embedding space actually has already captured some meaning or function similarity between different proteins, right? You can actually see clusters in this embedding space where protein ordered with different sequences. If their function are similar, they tend to cluster in the same space in this embedding space, right? That's very interesting to see that just modeling the sequences, you're able to actually capture some of the interesting function property of the protein. You can then use this kind of embedding for building lots of different kind of prediction models, right? Here, we tested actually now more than 15 kind of different tasks. I'm just showing 15 here. And for each row here, it's going to be a task, and the performance has been already scaled to between 0 and 100. What you see is this yellow-shaped diamond is almost always on the rightmost. It's getting the best performance. That's the 100 billion per chain model and fine-tuned with LoRa for different kind of tasks, including some of the tasks related to the structure of the protein, such as contact prediction, the fold classification. It's actually more than 1,000 classification problem, and also some of the kind of problem related to the protein functions, such as fluorescence ability, fitness of the protein, protein interaction, protein developability related problem as well, right? Very often you just see that this larger pre-trained model is also providing very good features for building some of the protein prediction model as well. So what's interesting is to actually look at how the downstream tasks also scale with the model sizes, right? And this is just one example figure, but we see similar scaling ability in other protein kind of prediction problem as well. What I'm plotting here in the horizontal axis is the compute, training compute. You can think about that is the modification of the model by the number of training data point you pipe into the particular model, right? So, and the vertical axis is the performance of the model. The larger, the better, right? If you look at this relationship in the local linear space, you actually see an interesting linear line like this. It looks like we still haven't hit the ceiling yet, right? It looks like, if you are able to either, get more protein sequence data, which is likely to happen in near future due to the sequencing technology, we're getting, lots of protein sequence data as long as you can get samples, right? And training with more computations, it's likely that the downstream task is going to improve further, right? So that's interesting. So that opens up the question is whether you can do more, right? It seems to be a quite certain way that we can get better performance for the downstream task, right? So here, what I'm showing you is some of these simpler tasks, but you can also look at the more complex tasks such as the structure prediction, right? And AlphaFold 2 is a very successful model in the space for taking the protein sequences and predicting the three-dimensional structure of the protein. But actually, the model is very complex in the sense that it's a very complex pipeline. First, you get your protein, and you go to the big protein database and find a similar protein. It's like a retrieval. It's like a search engine. You input your query protein, and you get a lot of similar protein. And then once you have these retrieved the protein, you apply deep learning model on top of it, which is called Inverformer and structure model. The original AlphaFold 2 model is actually consisting of 48 layers for this Inverformer, right? But with this pre-trained model, you can actually drastically simplifying the protein structure prediction model as well. instead of going for a database and search for similar protein, what you can do is to take the protein sequence and directly input these protein sequences into your pre-trained language model, yeah, and get embedding. And that embedding is going to feed directly to this formal model and then structure model for predicting the structure of the protein. Actually, by leveraging this pre-trained model, you are going to drastically simplify the structure prediction pipeline. And the deep learning layer can also be reduced from 48 blocks or layers to just one layer, right? And you can just chain this end-to-end with your structure and sequence input and output pairs, right? And what you will find is you get some structure prediction model. What I'm showing here is mostly for antibody structures, which are a family of protein important for immunotherapy, right? You can get the model for structure prediction just as accurate as AlphaFold 2, right? And yet at the same time, it's going to be much faster, right? Because the entire pipeline is going to be deep learning running GPU. You don't need to go for first searching for database and then go for deep learning, right? That's something interesting to see. You can actually benefit from this pre-trained language model to handle some of the very complex tasks in structural biology, right? So I mentioned earlier, the same model is trained for MLN and also GPT. Mass language model, trained model, pre-trained model is good for understanding prediction tasks. And generative kind of pre-trained model is good for generation. You can actually ask the model to generate protein sequences, right? And you can sample a lot, and asking the sample sequence to the AlphaFold 2 to get a structure, or the structure prediction model to predict the structure of those generated sequences. What you will find is you are able to generate actually similar structures as those structures you will see in the PDB database, right? Some of the standardized database, which people have already determined the structure, while at the same time, the sequence can be very different, right? In terms of the similarity between the structure, it can be as similar as 95% similarity, right? But when you look at the sequence identity, it can be very dissimilar. So that allows you to, in some sense, have a model to explore the potential larger design space and find novel proteins, right? And at the same time, it has a particular fold and function and binding kind of ability you want, right? Besides unconditioned generation, you can also condition this type of generative model on some other kind of information. For instance, here is an example where you can take a structure as an input. That's the desired structure you want to generate. A condition on that particular structure asking for what kind of protein sequences can actually allow you to get that kind of structure. So you can have a pre-trained structure encoder, use that to drive your conditional generation. If you do that, actually you will find that the model you built for conditional generation of the sequence, that's also called the inverse folding kind of model, can get much better kind of performance compared to one very famous model in the field, which is called protein MPMM, right? It has improved the ability to recover some of the existing protein sequence much better comparing to the protein MPMM. You can also look at the marginal distribution in terms of these sequence kind of positions you covered, and the pre-trained model is also going to give you a much better recovery, and in terms of making sense in terms of chemistry, right? Yeah. Now you see that you have this pre-trained model, which is on the one hand can support lots of accurate kind of classification model and regression model. On the other hand, it can also allow you to build some generative model. You can actually have these two type of model work side by side with each other to allow for more controlled generation, right? So take this pre-trained model, you can define the lots of prediction model to make a discrimination or prediction about what the property is for that particular protein. You can also have a generative model on the left-hand side. You can use these pre-trained task model or prediction model as some kind of reward functions or guiding functions to guide the generation, right? If you do that, you also find that you can actually generate those protein sequences towards a particular type of property or classes property you want, right? So here are just some example I'm going to skip. And we actually tried out this type of pre-trained model and conditional generation on real-world wet lab experiments. Here is an example where you want to actually design some kind of binder protein to a protein complex called the CD40L, right? The CD40L are trimers. That means that it's 3 proteins, they bundle together for a complex, and you want to actually design some proteins which can actually block the interaction between CD40 and CD40L. You want to block them, right? You want to design some kind of this blue protein here, which can block the interaction between this yellow protein and the green ones, right? So that means that you have to make sure your designed or generated protein is able to bind or recognize that particular interface between this green protein and the red protein. And also, this blue protein you design has to have a stronger binding affinity compared to the original green protein, such that it can compete with these green proteins to get a position, block the original kind of interaction, right? So, and we use this generative model to generate new protein sequences, and it has a different type of folds, and we tested out some of the good ones, yeah. So this design number 83 and 81, it actually has a stronger kind of binding affinity and results in better inhibition in terms of the blocking this original CD40L and CD40 kind of interaction. So nowadays, it seems like based on this type of generative model, and you are actually able to design some binders, right? And we are continually actually try to improve these property prediction and generation ability, conditional generation ability especially, to make sure that you have higher success rates of generating binders and proteins of particular function for more broad kind of problems, right? So here is about this pre-trained model for a protein, and you can actually get improved prediction model and also generation model. So I'd like to just briefly mention about another kind of pre-trained model in the space, which is more about the single cell gene expression data, right? So now it's more complex. It's not just about a single protein and protein sequences. It's about 20K genes that are expressed in human cell. You want to actually build a model to capture the dependency between how different genes are being expressed in the cell, right? So it's the expression of the gene is controlled by such a complex network. That's why there's some dependency between the expression level. But if you look at the data out there, and you can think about you collect this data, and each row is going to correspond to one cell, and each column is going to correspond to one gene, and you measure the expression of value of the cell, and you record these numbers here, right? Okay. So now you want to actually build some kind of pre-trained model to capture the dependency between the expression level of gene, and you can do something similar in this case by masking some of the gene expression level, and asking your model to recover it, especially just using the transformer architecture, right? But here you need to do something special in terms of designing your position encoding and how to handle a cell with 20K genes. It's like long sentence. You need to actually do something special in order for your transformer to be able to train effectively on such big data sets, right? So we designed a little bit of this transformer to make it asymmetric. Essentially, the model will handle those genes which are expressed very slightly or low expression genes and highly expressed gene differently for each cell. And then another thing we did to make the training more kind of suitable for the gene expression data is to take into account there's different sequencing read depth in this data. It's like you have different resolution for measuring the expression level of genes. Some lab, some kind of institute, when they're collecting the data, they are measuring in higher resolution. It's high resolution image. Some has a low resolution. So in order to actually allow your model to better harmonize, leveraging this type of different resolution, you have to design some kind of additional pre-trained task, which is super resolution kind of task, to be able to leverage the data and remove the heterogeneity in your data, right? So after you pre-train this model, we actually scale the model up to 100 million. It's already the largest in the field, but still not large enough. We find that again, the pre-trained model provides embedding which better differentiate different type of cells. And you see that the different type of cells with different colors that tend to be more clustered in this pre-trained latent space. So one interesting actually downstream applications for many pharmaceutical companies and many biological kind of research labs is, what if I have type of cells, either it's a cancerous cell, if I perturb it with a small molecule, or if I do some kind of genetic editing for that particular cell, what's going to be the change in the gene expression level? Is it going to move this cancerous cell to some kind of a healthy cell, right? Or if I want to kill the cell, what will be the best target, right? So then you actually very often in experiment do some kind of perturbation experiment. Take some cells, right, which might be cancerous, you put in different kind of drugs, or you do different type of genetic editing to perturb that particular protein associated with the gene. And you want to see what's going to happen to the particular cell. So you can actually leverage this pre-trained embedding or representation for a single cell and tie or integrate it together with some kind of representation for your perturbation. If it's small molecule, you can actually have representation for small molecules, and then have that embedding work together with this presentation for the single cell and to make prediction about cell responses, right? Again, with this pre-trained model, you're able to actually get more accurate kind of predictive model as well for both small molecule and also for CRISPR, like genetic perturbation. So I'm not going to go into details of this wet lab experiment. We also tried in some wet lab experiments. So hopefully, by giving you these two examples, pre-trained model for protein and pre-trained model for single cell RNA-seq, you get to see that actually the amount of unlabeled data for some of the biological problem is already reaching or close to the internet scale, billions of protein sequences, and growing exponentially. So there's really some opportunity for us to pre-train some big models, understand the property of these models, and use it for some of the important and impactful downstream kind of predictive problem and generative model problems in the biological space. But actually, there are a lot more questions than answers in the space. And I think people are just starting to explore using deep learning and large-scale pre-trained model for biological problems. So, for instance, it's not clear whether we can actually build foundation models for the biological problems, and whether we should build it based on protein sequences or actually just on DNA sequences, because DNA sequences are all the original of the information, right? And should we just build one for DNA sequence? And it can be actually used for RNA and protein and even cellular problems. It's not clear, right? What will be the best architecture for this type of deep learning model? And you need to be able to handle long contexts, because all these proteins, they have very long regulatory region and coding, non-coding region mixed together, right? So you have to look at potentially hundreds of thousands of positions in order to capture this type of long-range regulatory informations. And how do you actually go from genomic kind of model or molecular kind of model to a more system level model? So these are all fascinating questions, I think, and also offer us lots of opportunity to potentially build new models and develop new methods for it. But that's all I want to say today, and I just welcome you to look more into this type of problems and models, and there's a lots of interesting opportunities out there. Thank you.
Thank you very much for the very exciting talk. Other questions from the audience?
Thank you very much for your amazing talk. So there are a lot of interesting things, but I'm just particularly interested in the conditional generations. So it is the kind of, you are doing something like a reverse program. So how do you do that? So could you explain a little bit more details?
Right, so there are many ways you can do conditional generation. And one way is to, as I show here, it's something called actually inverse folding in the space of protein design. You take a structure you want to obtain, you are asking the question of what will be the sequence which gives you that structure. So you can condition on the structure here. You have an encoder of the structure, you get a vector, and you can use that to drive your sequence generation model. You have to use some of the structure sequence pair to fine-tune it, to generate. The second way is actually you have an unconditional generation model, like here, but you have some property you want to obtain, for instance, binding affinity or stability of the protein. Each one of these properties is going to correspond to one predictive model, right? So in a diffusion model, you use that classification or predictive model as a guidance function, right? Here, actually, you can use this model as your reward function to try to actually fine-tune your sequence generation model towards the high reward region. Right? There's some method called the REST or all these different type of reinforcement that you can actually use to fine-tune your generation model towards particular property. Right? Yeah. There are multiple ways. Whatever ways you can use for, in some sense, natural language. In some sense, you can think about how do you actually transfer to this scenario. Right?
Interesting. Thank you.
Other further questions? Yes, thank you for this very fascinating talk. Nowadays, in the area of large foundation models, people are interested in aligning different modalities with a language model. So as for the biometric foundation models, we just think it's necessary to align the bioinformatics with the language.
Yeah, very good question. I think definitely there's a need for alignment, but in this case, it's not aligned with a human value or human opinion or something like that. It's more like aligning the model with wet lab experiment, the kind of property you're going to actually measure in wet lab experiment or some of the physics kind of property, right? So at the moment, this pre-trained model, for instance, for the protein is more on just the sequence, right? And looking at the, in some sense, the co-occurrence ability, probability of the amino acid in the sequence, but it may not fully kind of understand physics, for instance, molecular dynamics and some of the energy between these different actants, right? So how do we actually leverage some of the physics information to guide our pre-trained sequence language model to becoming something better? So we don't know, right? But that's a very interesting kind of research question. Yeah.
Are there further questions? Okay, if not, let's thank Sam again for the great talk.
Right.